Good afternoon, everyone, and welcome to a special edition of our Zoom webinar series, You, Me, and PD. I'm Vince Kinsler, the Executive Director of the Parkinson Association of Greater Daytona Beach, and pleased to have our guest today is Carley Rusch. We refer to her as a Neurology Dietitian there at the University of Florida. And Carley's been with us previously, and we're delighted to have her back. We're going to be talking about it was maybe 7 months or so ago that this paper had been published. And we're going to be talking about that. One, what led up to this, the paper, the study basically before the paper came out, and then what was going on subsequent to the paper, which really I think just concluded here in the last couple of months. And then what plans that we may have in the pipeline for continued research in this area, because the findings were quite optimistic, as we will see. Now, I will say, even though the paper had been made available to everybody in attendance today to download, print off, myself included, it was now what we might refer to as easy reading. So we'll be getting into some of that terminology, and Carley she's got a slide presentation for us today that will kind of walk us through so that we have a better understanding of what it was that we looked at. Like I said, it wasn't like reading a romantic novel or anything like that. It didn't go down as easy as candy, we should say. But with that, I'm just going to go ahead and turn it over to our guest today, Carley Rusch. Please join me in welcoming, and let's get to it.
Yes, thank you for having me. I will say that reading a lot of other research articles is not easy for myself either, as someone who writes them. So I can appreciate the difficulty sometimes it takes to, digest and learn the information. But that's why I'm passionate about teaching research methods. I taught 2 semesters of a graduate course here at the University of Florida to dietetic students on research methods as well. But yeah, so I guess if you'd like, Vince, I can kind of just go over just like a quick background on what are the different types of research studies that you might see out there. How does that sound?
Yes, that would be helpful, because as an example, this one was a single arm. I've not really heard of that, but then again, I'm sure it's very common terminology in the field of clinical studies. I'm more adept at the blind or double-blind studies. Right. So maybe some differences there, but whatever you can tell us, obviously, is going to help.
So yes, the design of my study was, so it was a 5-week intervention study, but it was a single arm, which just means pilot study, which just means that there were only one group doing the same exact thing throughout the whole study. And because it's a pilot study, that just means that we were collecting preliminary information to help inform future research in the future. But just to kind of give you an overview of research study designs. Oh, let me switch my screens. Swap. Okay. So just to kind of like backtrack like a number of steps, because I know a lot of us don't have a research background. I did it at one point too, so I can understand how complicated it can seem. So when it comes to study designs, we basically have 2 categories. We have observational studies and intervention or experimental studies. Observational studies just mean that we are observing information or in data, right? Intervention studies mean that we are providing an intervention or we're completing an experiment in a study. So for observational studies, there are a number of different types of observational studies that we might have. One of those types is called a case series or a case study. That just means that you're describing a unique case that someone might present with. So for example, if you have Parkinson's disease, maybe you've gone to your neurologist and they're like, you know what? I believe you have Parkinson's, but your symptoms are more of an atypical presentation, right? When we're describing diseases and conditions, we as healthcare practitioners, we try to, simplify and group diagnoses and conditions together. So case series and case studies are really helpful because it's people in the research community describing when something doesn't fit within the box, right? We say, we believe it is this, but it doesn't fit within that diagnostic criteria. It doesn't fit within this box of what we know. So this is a very preliminary initial finding that someone might report on. So that's what a case study or a case series means. A case control study means that you have someone or a group of people with a known diagnosis or condition, and you have another group of people without that diagnosis or condition. And all you're doing is just comparing differences between them. So for example, when it comes to Parkinson's, a lot of case control studies, for example, in the microbiome community are, if we have someone with Parkinson's and someone without Parkinson's, what are the differences in their microbiome signatures look like? So it's observational. We're not providing them anything. We're just comparing differences at a snapshot in time.
Even though this may seem rudimentary at this time, because I'm certainly familiar with the term microbiome and microbiota and what have you, can you just give us a lay term on microbiome?
Yeah. So the microbiome is essentially the community of bacteria, fungus, viruses, even dead DNA that's just within your GI tract. So you have an oral microbiome. So you have different bacteria that are colonized within the mouth, and you have a small intestinal microbiome. There's not a lot there, but you have a different number of bacteria, viruses, et cetera, that are there. And then you have what we're more traditionally hear about is the microbiome within the colon. That's the largest density of species of bacteria, funguses, viruses, and they all live in a community. So we have beneficial bacteria, and we also have more pathogenic bacteria. And to have, optimal health, we need a balance between, more of those beneficial members versus the pathogenic members. So it's just one of those hot topics in the healthcare community because we know that the microbiome can influence a number of different diseases and conditions, including people with Parkinson's.
Now, I've read that there's something like 100 trillion microbiome in the gut, in the average human. Yeah. So, I mean, trying to determine what is good and is bad, that gives new meaning to a needle in a haystack, I would imagine. But we can get to that further. Thank you for that.
Sure.
Yeah.
Yes. Yeah. We'll talk about the complexity of the ecosystem a little bit. But yeah, so just to kind of circle back to the study design. So, I talked about the case control. So you're just looking at someone at a snapshot in time and saying, what's different between someone with Parkinson's and someone without Parkinson's? A cohort study is where you might prospectively follow someone over time. So for example, there are a lot of cohort studies within the Parkinson's community. I've heard, I'm blanking on the names of some of them that are coming, that I can't think of, but I know UF Health is a part of a few of them. And that's where you might have gone to a movement disorder clinic and they asked you to consent to be part of a database where they just follow your information over time. And that's where researchers can look at, over the course of 5 years, why did, for example, someone's motor symptoms progress more than others, right? And identifying some preliminary data to answer some of those types of questions. In the nutrition community, cohort studies might look at people, for example, heart disease is an easy one to look at. Well, not necessarily easy to look at, but it's an easy example to use. So if we look at people over time, and let's say we look at their fat intake, and that in a cohort study, we might look at who were more likely to develop heart disease and who weren't, based on the food that they were eating over that 5-year time period, let's say. So it's still observational. We're not changing anything. We're just collecting data, but then comparing why people develop certain diseases and conditions over time versus others. And then you have some other observational studies like review articles. So a narrative review or an expert review just is maybe 1, 2, 3 or more people that come together and they just review a certain topic. You have a systematic review where it's a little bit more methodical in terms of the steps. A narrative review can just be very broad and you kind of determine what you want to talk about and what you don't want to talk about. A systematic review, it's a lot more methodical, where you're talking about what you find, how you found it, and what information did you find from there. I won't talk too much about that one for now. And then switching gears to intervention studies, which I know a lot of people within the Parkinson's community want to hear more about, is what are the new, drug trials and therapeutics and different interventions, like whether it's exercise, diet, nutrition that we could be doing. We have, when it comes to experimental studies, we have non-human laboratory studies. That's what you might find mouse, rat, monkeys, even like dogs and pigs. You might see some researchers using those types of studies. The purpose of doing that is so that way we can look at different mechanisms and really explain why does something occur that might not be ethical to do in a human. Human studies are going to be some of those randomized control trials. And you might have what's called a parallel design or a crossover design. So you might see this was a double-blind, placebo-controlled parallel study. That just means that someone was assigned to one of 2 or 3 groups or more, and they just did that intervention over the course of the study. A crossover study is really interesting because you actually will be randomized into both groups. So for example, if you have a placebo and an intervention, you'll get randomized to either start with the placebo or start with the intervention first. You have a washout period, and then you'll switch to the treatment that you weren't on. When you're with a parallel study, you're just comparing data between those 2 independent groups. So the people who are on the treatment and people who are on the placebo. With a crossover design, you got both. So you actually get to control some of the individual variability because you're comparing treatment versus placebo within that one person. Versus
question along that line, is both of those parallel and crossover, those since there are 2 groups or many groups, if you will, that is are one of them always a control regardless? Is somebody always a control in those?
Yes. So when it comes to randomized controlled studies, there's always going to be a control in them, just by the nature of the design.
Gotcha. Should have read that first. Go ahead.
No. So sometimes, there are a bunch of different limitations, whether it's time, money, resources, right? Researchers would love to always do human randomized control studies, right? That are, double blind, that limit bias and really help strengthen the results of the study. But you have those limitations of time, money, and resources. So sometimes we might not be able to do everything that we want, especially if it's on a topic that hasn't been well studied yet. So for my study, I'll kind of bring it back to the name and the title, right? All right, let me just exit out here. We did a single arm pilot study. So it was a very small study, 8 people, not very large. And because the study was so small, we didn't have enough people to really say, okay, 4 people do an intervention and 4 people do a control yet, because these is just very baseline findings. So we just want to put everyone on the intervention and see, do we see any changes, right? That's why it's called a pilot study. It's just the beginning. It's not that gold standard yet. And it's a single arm because there's no control group. Does that make sense?
Well, sort of. In this case, if I read it correctly, there was really 16 people involved.
Yes. So this study was very unique in its design, which is why it was written up as, you could see up here, a brief research report, because the study design has its own limitations to it. So it really is a case control study because we recruited people with Parkinson's and their partners, and we compared their samples at baseline. So that case control, we looked at differences. But then because it's a pilot study, we put the people with Parkinson's on a Mediterranean diet intervention and just see, okay, if we know this is different at baseline, what happens after the diet intervention? And by doing it in a small group of people, that allows us to target our resources, time, and energy to information that we're more likely going to see change from in a larger study.
Is that into the way you just described that, Carly? Is that why there was no post 5 week, only baseline for the control?
Correct.
Because you really didn't want to study those people, even though it kind of would have been interesting, I think, but.
Sure. We just wanted to see what was different at baseline.
To begin with.
And if those differences changed after the intervention.
But you weren't interested in the control.
Nope. Because we know we've seen, here's the thing. There's already information out there on people without Parkinson's and how, if they follow a Mediterranean diet, how the beneficial bacteria in their gut changes. There's already that information out there.
But ours was specific to constipation.
Exactly.
Beautiful.
Awesome. What other questions would you like me to dive into about the article?
Well, before we get into the article, I guess,
Sure.
And getting back to some of the terminology, again, I mean, there's plenty of terminology in there that's going to be quite nebulous to most of us, but some that comes up quite frequently throughout. And we already asked about microbiota, and we got that covered, the microbiome. The term intestinal permeability.
Mm-hmm.
Term that for me.
Sure. That's a great question. So intestinal permeability essentially means the ability of different bacteria and molecules, whether they're pro-inflammatory signaling molecules or anti-inflammatory signaling molecules, to permeate the intestinal barrier of the GI tract. So, okay, what does that mean? So when you think about the GI tract, and as you have food and stool passing through, it goes through basically like a tube, right? And on the outskirts of that tube, you have a very tight lining of cells called epithelial cells. I even have a picture of it here for you. Let's see. Awesome. So along the GI tract, you have these epithelial cells that are stuck together really tightly, as you can see here. And they're stuck together very tightly by these squares here, which is what we call tight junction proteins. Okay, so as stool is passing through, and just even when it's not passing through, and it's just, your GI tract is just kind of hanging out, waiting for the next thing to come by, you have bacteria that are floating around within the lumen of the GI tract. There's even, I don't have it here because this is just a very basic rendition of intestinal permeability. But even between these cells, you even have a mucus layer at different thickness, and bacteria will actually live within this mucus layer too. And these are more beneficial bacteria that live here that provide benefits to us as humans. But, let's say we have bacteria that are floating around. Maybe we have a little bit more pathogenic bacteria because I have a lot of GI symptoms like constipation, gas, bloating. And some of those pathogenic bacteria are now kind of thriving in my microbiome environment. Well, those bacteria, as they're floating around, may send out some signaling molecules, which you can see in these little green circles here, that say, hey guys, let's ramp up inflammation. When that happens, when we're increasing inflammation within the GI tract, some of those tight junction proteins start to go away. And as they're what we call as scientists downregulated, meaning we're just not producing as much to line those epithelial cells and make them stick together. When we have less of those tight junction proteins, we start to get an opening of the barrier, right? Remember I said it's a tube, we have a barrier, right, for things to pass through. As that barrier opens, or what we call leaky gut, that allows for more passage of bacteria and other signaling molecules that can really ramp up that inflammation and cause more GI symptoms. So that's what the term intestinal permeability or leaky gut refers to. Okay.
Thank you.
Yeah. And so as researchers, we have this question of, okay, how can we study intestinal permeability in people that's not as invasive, right? Because if you think about it, how else would we study this? We might have to, maybe that might mean sticking a sigmoid, like kind of similar to how you get a colonoscopy, right?
But not quite as far.
Not quite as far, at least. And in sampling the lining of the GI tract where we can measure these proteins, right? But again, it's a little more invasive. Not a lot of people are going to want to always sign up for that type of study. So can we do something that's not invasive, but still can measure what we're looking for? So about, I think it was about 20 plus years ago, there was this method of looking at intestinal permeability in humans using a sugar cocktail and measuring those sugars in someone's urine over time. So how it works.
Can I ask a question there? Because I found that to be quite interesting, why the urine. I'm just wondering if there's a breakdown in that sheath, if you will, that membrane, those little squares that started to separate and allow the bacteria and the other stuff that we don't want into the leaky gut or from the leaky gut. Does that now get into the system whereby if you put the sugar that's got very low weight or whatever, it escapes very easily? So if it's escaping easily, it comes through the urine. And is that why? Or am I?
Correct. Because I didn't have a figure on there, but from the barrier, from the lumen of the GI tract, then you have the barrier cells. You have little capillaries, veins that go back to the liver to get reprocessed, and then back out, and then the blood is filtered through the kidneys, and the kidneys excrete any excess products out from there. Mm-hmm. Gotcha. And so what's great about using a sugar cocktail is that you can actually measure these sugars in someone's urine.
Yeah, yep. I saw something in the study too. Forgive me, I'm kind of, and I'm going to jump forward a little bit, but it had to do with a product that you were using that, or was part of your cocktail that showed up to be not the correct product.
Yep. This is a great example of why you should be very cautious of where you purchase any supplements and food products from. Supplements in particular is because just because something is packaged and says it is this on the label doesn't always necessarily mean you might get that in the actual product that the manufacturer sends you.
So even you research scientists, the University of Florida can get taken like that, huh?
Exactly. And, really, like, this is the beautiful thing about research is that as researchers, we are ethically required to report if something wrong happened, right? And so that is exactly what you read, Vince. We messed up and something wrong happened where the sugar that we purchased was not the sugar we intended to give. And luckily, we have an amazing team of scientists who use a method called mass spectroscopy, where they can actually look at what sugar is in the urine and use databases to compare what we actually give. So That sugar that we were supposed to give was a rhamnose, is what it's called. And it turned out to be neotame. All of these sugars here are sugars that are approved by the FDA to give to humans. It would have been more problematic if we found something that is not approved to be ingested by humans.
Yeah.
So luckily,
as long as you had your waiver signed, forgive me.
Yes, it is something that we did have to inform our participants that happened afterwards. But this is the beauty about transparency in research, right? We were transparent and we said, hey, this happened, right? And this is how we figured out how it happened.
It looked highly recoverable based on what I read. So
yes. And so I can kind of explain the sugar part now too. So when it comes to measuring intestinal permeability, you're like, okay, we're going to give sugars, we measure it in the urine, but how do we know what we measure in the urine means what? Because remember, like I said, we have a GI tract that goes from the top of our mouths all the way down to the bottom, right? And when it comes to constipation and the microbiome, we're particularly more interested in looking at the permeability of the colon right here, right? But we also want to know, permeability at other sites too. So how can we distinguish between the two? Well, that's why we use different types of sugars to distinguish between the two. So, I just have it on here. If the barrier is compromised. Okay. So the first sugar that we use is called sucrose. It's actually a commonly found in table sugar that you can find in the grocery store. And we give pure sucrose to people. And we know that sucrose is rapidly digested and broken down. In the duodenum. So we know that sucrose, particularly immediately after ingesting it. So we have people, I'll backtrack a little bit more too. We have people collect their urine for 24 hours from the start of that, when they drank the sugar, to 24 hours after they drink the sugar.
So is that one of those things where you give them a gallon container?
I actually have a photo of it here.
Yeah, I've done that. I'm like, there's no way I'm going to fill this up. And by the end of that 24 hours, I was getting frightened that I was going to overload.
I had one spouse who was an overachiever and she filled up 3 of these. Yep. We gave them urine hats to make it a little bit easier
little cup, hat and cup.
Yeah. And so, people are eating within this time frame, right? So how do you distinguish the sucrose? And I just said it's commonly found in foods that we might have to eat. So how do you distinguish, is the sucrose that I found in this 0 to 5 hour time frame actually from my sugar cocktail, or was it from something else that someone had, right? So that is why the first 5 hours, our participants fasted in the study after consuming that sugar cocktail. After 5 hours, so you'll see like this 5 to 24 hours, they continued to eat, but we knew that it takes about a couple of hours for food to pass through the digestive system. So we knew that the sucralose and erythritol, for example, that we were measuring in the colon likely reflected more, especially within the 5 to 24 hour time frame, reflected more of colonic permeability, right? It would, in a perfect world, we would tell them not to eat at all, right? But we no one's going to sign up for that study.
My grandkids might.
Yes. So we use different types of sugars based on what we know about their physiology, how they're digested and broken down. So sucrose, we know, is broken down rapidly in the duodenum. So that represents gastroduodenal permeability or leaky gut. We know that lactulose is rapidly digested and absorbed within the small intestine. So that represents small intestinal permeability. And sucralose is more rapidly absorbed. It really shouldn't be broken down at all and digested at all. But if some is, it's going to be more so within the colon. And so by using these time points and different sugars, we can determine different sites of permeability. And you can see here we have different ratios. And we use ratios, for example, lactulose is a very large sugar molecule. rhamnose and erythritol are very small sugar molecules. So we use these ratios to help minimize the effect of, for example, someone's transit time throughout the GI tract might affect how much of these sugars they actually absorb or not. And they also help control for kidney function too. Does that make sense so far?
Yes.
And when it comes to like what we've known from previous research, leaky gut and permeability using this method hasn't really been extensively studied in previous research. Only about 2 studies, and they were done 10 years ago. And before that, the first one was done probably about 15 years ago. So it's been quite a while, right? So, just because 2 people have found it might not, in a very small sample size, might not mean that's the case for everyone. So, it's important to reconfirm what has been found in previous studies as well. So that's why, another reason why we wanted to have a control group too for this section. So what we found was nothing. So just taking a look at this table here that's in the paper. So at first we compared people with Parkinson's versus control, so that case control study at baseline, right? And I'll just kind of draw your attention to the ratios here, because remember I said the ratios are a better measure of intestinal permeability because we're controlling for transit time and someone's kidney function. P-values in research just help us distinguish, is our observation, did we find this by chance, or is this a very unique finding that we found? So ideally, researchers set a cutoff value of a p-value of 0.05, meaning that that finding is significant. So you can see here for both sucrose, lactulose, erythritol, sucralose and erythritol for the colonic, and then whole gut permeability, we didn't find any significant differences between them at baseline, right? You could see here, you're looking at the controls, right? They had about 9.7 micromoles of sucrose versus 11.5 micromoles in Parkinson's, right? But how do we know that this is a significant finding? That's what this p-value helps us determine. So really, it's not significant. It's what we would expect in a normal observation, okay?
Is that where that plus or minus comes into play?
So this plus or minus actually, so these are averages here. So this plus or minus is the standard error of the mean. It's basically taking the standard deviation and controlling for how many people that you have in your sample.
So somebody in that group actually had 2.9 greater somewhere maybe?
Correct. Yep. You might have someone that's about 3 micromoles above it or 3 micromoles less of this value. Correct. Okay. That's what that means. All right. But, we wanted to look at these single sugars too and see, okay, was there any differences there? And that's maybe why we didn't see differences in the ratios. So we did find that within the small intestine, so that's 0 to 5 hours, the lactulose and the erythritol were different after a Mediterranean diet. Not really at baseline between the control groups, but when we gave them a Mediterranean diet, these values changed. Well, why is that? One of the reasons we suspect is maybe because their transit time changed, right? So if we gave them a Mediterranean diet, they consumed more fiber. Now they're having more frequent bowel movements. They're less constipated. And maybe there was more exposure of the sugar to the lining of the GI tract. And that's why we saw a significant difference there. That's one explanation we have for it. That we might have. What that might be the explanation or a prospective explanation, but what significant value might that otherwise have? I mean, is there an explanation for that, or is it not anything alarming regardless, other than the fact that it was significant as far as its difference? It's just saying that it's significant as far as its difference.
So it's not either good or bad.
Exactly. We don't know. We don't really think it's good or bad. Because you could see these values. For example, we have at baseline, on average, people had about 12 micromoles of lactulose that was excreted from their small intestine. After the diet, it was 21. So it's like, oh my gosh, like, is their GI tract more permeable? Right.
Right.
But when we look at the ratio as a whole, well, both of these values changed, right? Because remember, we're using these values to help control for motility of the gut. And if the food is moving through faster, maybe it's being exposed more or less. It's hard to say because we're not in there either, right?
Well,
we're just measuring things after.
A question along that top box. I mean, so the controls actually consumed the cocktail.
Mm-hmm. Yes.
Okay. All right. So that's why you were not all excited about the increase.
Correct.
Because on the control side, they had similar advances of some sort.
Mm-hmm. Mm-hmm. Yep.
Thank you.
Yeah. And like I mentioned, we really think that these differences in the single sugars were more related to the transit time versus the actual permeability of the GI tract, because everyone is going to have some degree of leakiness, right? We anticipate that. So that's why you want to look at things that's kind of like a greater scale and on a relative way. And that's where ratios kind of help come into play here. So this was the more exciting experiment that we got to do, and we really didn't find anything very significant there.
Looked exciting for a second.
But you know, what's really important about pilot studies and pilot research, like I mentioned before, it helps inform the direction of future studies. So that way you can dedicate that time, money, and resources to, you're not just, going down a rabbit hole where you're not going to see any, discover anything new, right? Yeah.
Go ahead. I'm sorry.
I was just going to say, this was a very expensive experiment. And so for a larger study, we're like, okay, maybe it's really not worth doing this in a bigger sample if we don't really anticipate we're going to see any results there.
It seems as though, based on what I've read and what I've heard here yet this afternoon, is that there's been maybe a couple of studies, now not certainly focused on the constipation component, but regarding some synonymity of different things regarding the diet and what have you. But I kind of wanted to move forward along those lines in the sense that what number of human beings in any kind of a clinical study where you're going to get that impact. Okay, say right now you're seeing some synonymity. You're using stuff from previous studies that are showing up consistent in your study. So you're talking 8 people here.
Yeah.
So moving forward, is it 800, 8,000? Is it based on the Food and Drug Administration's? If you're looking at a pharmaceutical as an example, how many study people do they need in order to get things looked at and say this? Of course, there again, it's tied back to the cost, is it not, of the study?
Yeah, no, that's a really great question. And that's why we have actually statistics. We have statistical methods and models where we can actually take known previous data and determine, okay, if we anticipate this type of effect. So for example, if I think that with my experiment, colonic permeability is the ratio of it is going to change. Let's just throw out a number and say by 1, right? I can take those means and standard errors and standard deviations and impute it into a statistical model and say, okay, this is what I know from previous data. And this is the anticipated change I would want to see for this outcome. How many people would I likely need to see? Would I likely need for my experiment to see a significant effect that I'm looking for?
Wow. And so there's an app for that? Is that what that is?
I don't know if there's an app, but
it sounds like there's something out there.
Yes, but there is a statistical method for that. It's called a power analysis, if you ever hear about that.
It's called mathematics, right?
It's a lot of mathematics and statistics. I mean, I couldn't even explain the equations probably very well, but I think we did, I don't know if we actually looked at the numbers, but based on these differences here, it would definitely be a couple hundred people.
When you do that couple hundred people as an example, mm-hmm. I would imagine that it's almost exponential to the cost to some degree. Exactly. More people, more cost, right?
Exactly. Right. So,
yeah, but if you could cure so many people just through food, right?
Mm-hmm. Yeah. I wish. It is worth it. I wish, funding agencies saw it that way too. But yeah, so that's what a lot of researchers will do before actually even initiating a study is take a look at previous data, think about the question of their project and compute it into a statistical model. And that's how we actually determine how many people do we need for a research study.
Gotcha. So this is not really a research study, so to speak. It's a brief, what was it called?
Well, it is a research study.
I understand that. It's a
pilot study.
Thank you.
Yep. It's a preliminary pilot study. The purpose of this is to look at, hey, what is different? And if we give an intervention, does anything change? And if something changes, can we confirm that in a larger study?
Beautiful. Mm-hmm. Thank you.
Yeah.
Sorry to take you forward, but.
No, you're good. What other topics, I guess, from the paper?
Did that little glitch, if you will, on the L-rhamnose, did that affect the study at all? I mean, did it delay things, or did you discover it before you made the cocktail and then you had to tell people about it? Or I'm just wondering.
Yeah, that's a good question. So we discovered it after the study. So with human studies, oftentimes you cannot analyze your data until you've collected all of your data. And that's just to help minimize variability. For example, the person analyzing the data, right? You don't want different people using different types of methods and skills, and then trying to compare your data to each other. So we discovered it after the fact, after the study has already been concluded. I would say it may have delayed it a little bit. It just may have taken like a couple more weeks.
Because they needed to re-extract the value somehow, but they knew that the product they used was okay. They just need to look at it a little differently.
They basically just had to run it's what is called in chemistry an unknown experiment, where you say, this is the compound that I see, and measuring these are the characteristics of it. What is it, right? It's actually an experiment you would do in like a chemistry class in college. What unknown do I have? And once they figure out like, okay, I think it's neotame. Now they actually buy neotame and compare. Is it actually neotame? And it was.
Okay.
Yeah. So, but it really, what we decided was, since it was rhamnose, and rhamnose was that smaller sugar that we use to control for gut motility and kidney function, we had another smaller sugar that we gave them, erythritol. So we compared, instead of lactulose and rhamnose together, we compared lactulose and erythritol together because erythritol and rhamnose share very similar properties.
And going to the paper, and I'm going to go ahead and refer to a page number because I actually, going through the pages, I had to assign numbers to different categories just so I wouldn't lose myself. But on page 3, where it was laboratory assays, and I think on page 4, which was still DNA isolation. One, I mean, those terms, those components, if you will, to the study, I found to be wow. I mean, when I say wow, I mean I had no clue what the hell it was I was looking at. But so I'm going to just kind of throw this out there on the laboratory assay, were these just generally accepted standards or common equipment used for those type of analyses? Is that what those are?
Yes. And
Okay.
so when it comes to research, like writing research methods, when it comes to the whole research process, we want things to be replicable, right? So by listing out the equipment that you use, what settings you might have done on it, how you used it, what databases you might have compared it to.
Self future researchers, yes.
Exactly. It's really more so for, number one, to confirm that to, let's say, the journal reviewers that you did the appropriate method. And number two, so that way future people could go back, see how you did this experiment, and it replicate it if needed. Gotcha.
Now, the DNA side of it, even though it also, to me, was quite nebulous, to say the least, very technical reading, certainly, but not understandable to any human that is not a research scientist, I would surmise. But let me ask, what is it specifically about DNA that you were looking at? What did that have to do with anything?
Right. So we were measuring DNA, more specifically bacterial DNA, not human DNA. So we were looking at bacterial DNA in the stool samples that people provided. So I even have a stool kit here. So people at the beginning of the study, for both the controls and people with Parkinson's, and then after the intervention, after the Mediterranean diet, people with Parkinson's, they gave us a stool sample and shipped it in the mail overnight to us. They put it into little tubes that stabilize the bacterial DNA in the stool. And then I didn't do it myself, but another laboratory group that we collaborated with extracted the DNA from the sample to look at and compare what bacteria is there. Because if we know the DNA in the stool, we can actually use databases to predict who is there and who is not there.
I noticed too that they pulled out certain like strands or things like that just to try to keep things as consistent and pure as possible. From what I heard. Yeah.
Mm-hmm. Yeah. So I have a picture here I'll show you. So when it comes to microbiota analysis and sequencing, this is the most common way of looking at the microbiome that we have available to us. That's quick, fairly affordable in the research community to do, and it can give you some very interesting results. So, once someone gives us their stool sample, we can extract the DNA and amplify the reads from the sample there. And for those that don't have a biology background, some might, some not, but DNA encodes in different sequences. So you have different sequences of amino acids, and that DNA predicts, we know from databases certain types of bacteria. So you might have a sequence of a G, an A, a T, a C, a U, etc. Right? So once we've amplified and we've determined the sequence of DNA we think is there, we actually will enter in these sequences into a database of bacterial DNA sequences that we know and group them into what's called operational taxonomic units, or OTUs. So sometimes there's different variations between different types of bacteria, right? Bacteria, there's different, there's a phylogenetic tree, and they can go by a phylum, a family of bacteria that share similar properties, a genera, a species, and a strain. So when we're looking at, for example, at the phylum level, who is there, we might group those OTUs all together. Same thing with the genera. These genera represent this bacteria, right? And then all the way down to, most of the time it's species, and then some of our databases can give us strains. we don't have everything in these databases. We're still discovering new bacterial DNA sequences. Even now, we're even sometimes changing the way certain bacteria is classified. So it's a very complex ecosystem.
Yeah. Well, let me ask you this about that. Was there anything that you discovered that is now going into the database that had not previously been there?
No, not necessarily.
Not what you were looking for, right? I mean, if you're doing your input, looking at what is already available, did anything kick back and say not available?
Oh, yeah. There was plenty of DNA reads and sequences that we got that we just couldn't identify it. Or
that's kind of what I was wondering, if by chance you discovered something. But
yeah, so no, but I would have had to
different research, huh?
It's another microbiology experiment. But so you might get, okay, I know this OTU belongs to this family, but I don't know the species and the strain, right?
Gotcha.
So sometimes you'll get like kickbacks like that. Once you know all the different OTUs and what families, species, and maybe even strains they belong to, you can look at their abundance compared to the DNA, the total amount of DNA that you have. So if you know I have, for example, we can use simple math. So I know if, let's say I have 10 DNA reads and 5 out of 10 of those DNA reads belong to this specific OTU. That means I have a 50% abundance of this OTU compared to the entire sample that I collected. So that's what relative abundance means within the paper. You can use that information to create a phylogenetic tree, which some microbiologists and biostatisticians might do. We didn't do that in this study. And with some even more methods and technological experiments, you can even predict and looked at the genes that these bacteria express as well. So I have this kind of down here. So what I described to you, and a lot of microbiome research does, it describes what organisms are present. We just know who is there and who is not there, right? That is what the 16S rRNA microbiome sequencing tells us. What it doesn't tell us is what they are doing. And that's a major limitation that people have to recognize when it comes to microbiome research, is this method in particular only tells us who's there, but not exactly what they're doing. And so you might see, Vince, I don't know if have you heard of like these like stool sample companies where you send in their stool sample and they tell you what your microbiome is?
Oh, yeah.
Yep.
Yeah, I've heard of that.
So that they're using this method to tell you like who is there and who is not there. The problem with those types of services, while, as a researcher, I think it can be kind of cool to know personally on an individual level who's there and who's not there. What do you do?
Yeah. Do you take a probiotic that says they'll give you 10 million probiotics guaranteed, right? Well, there's 1 trillion in your gut. Who knows if it's going to match, right?
Right. So that's, and that I can really go down the rabbit hole, but it's another, topic of discussion. But, that's the limitation of this information that I think is important for people to understand if you're ever, finding yourself more interested in the microbiome and diet and Parkinson's. Is right now we only have a good grasp of who is there and who's not there, but we need more information on what they're doing.
Yeah, it's compelling and curious, but it probably doesn't have a great value.
It's very preliminary value. It's not diagnostic. It's or something we can use to treat necessarily just yet. But yeah, it's very preliminary. So that's why, and I'll just kind of like switch gears. You'll see sometimes a figure like this in a table. So on here, we just took all of those single OTUs that we found significant differences between control and baseline, as well as significant changes after the intervention. and we plotted them on a heat map. So this heat map represents abundance of who is there and who is not there. And we just compiled the most relevant ones here. So what's really cool about a heat map, you can compare. So for example, I have it broken up into different slices. So section A, the top here, this is OTUs that were more prevalent in our control group versus Parkinson's. So you can see here, a lot of hot buttons, right? And not very many in people with Parkinson's.
Well,
right?
that first column to the left is control.
Correct. Mm-hmm.
Okay.
And the darker the shading, the more prevalent that OTU is in the sample. And so what researchers can do is look at, okay, who is there and who is not there? So for example, one of the more, if you've ever heard any of my other talks for like the Parkinson's Foundation, I love to talk about the some bacteria that are kind of get a lot of attention in the research community, which one of them is called Faecalibacterium prausnitzii. Faecalibacterium is the species, the genera, prausnitzii is the species. This one gets a lot of attention because it's a known beneficial microbe. The more we have of it, the better for our immune system and our gut health. So you can see here, there are single OTU reads of that F. prausnitzii that are more prevalent in the control group versus Parkinson's.
How do you know what little box relates to the line item?
Yeah, it's basically just kind of going across here. So for example.
So it is just across the board, basically? I see. So it is, okay.
Yep.
It's just an extrapolation. Got it.
Exactly. And then so you can see here in B, you have OTUs or, single strands of DNA that are more prevalent in people with Parkinson's compared to people without Parkinson's, and these columns here, right? So what are some that might be more prevalent? Well, sometimes we might see some more less beneficial bacteria. I can't see. I know these can be really hard to read. I won't spend too much time on it. It's very hard to see here. But anyway, some of these bacteria can kind of function between pathogenic and non-pathogenic. We're still kind of figuring that out in the research community. But then you can also see here for on C, the bottom, these are the bacteria that were decreased after a Mediterranean diet intervention. So
that's just Parkinson's only, though.
Correct. Only in Parkinson's. Mm-hmm. Yep. So this is just kind of to help inform, future research and maybe what are some species you want to look at or not.
Can we digress for a moment?
This is Elaine.
Hello, Elaine.
I have a quick question. Apologies for jumping on late, Carly. I had another thing run late, but this graph is really interesting, especially when I look at the top panel where you have, the control on the left, and then it looks like with the Parkinson's disease from baseline to intervention, you do see change, right? Like you can see that there are more there at after the 5-week intervention. And it just makes me wonder, what are your thoughts on the fact that it was just 5 weeks, say, versus like, a year or 2 years or even a lifetime?
That's a good question. So we know when it comes to our microbiome, it can shift fairly immediately. If you fast for a day and don't eat and you compare your microbiome signature before and after just a day, you'll see differences there. So when it comes to interventions and research, typically we might do something over a couple of week time period. So this was a 5-week intervention, but we probably would see bigger differences, assuming people followed the diet, strictly and appropriately, if they were to follow it over a longer period of time, let's say 6 months to a year. And that's just because when you think about the ecosystem of the bacteria, it takes some time for certain bacteria to what we call engraft, where even if you stopped following the diet, let's say for like a day, right? Or probiotics are an easier example. Let's say you stop taking a probiotic for a day or two. You won't find that bacteria in your stool anymore. But let's say you took that probiotic maybe a year. It has a longer availability and chance to actually engraft and thrive in that new environment it's been introduced to, where if you stop taking that supplement, that probiotic supplement for a day or two, you'd still probably find it in your stool because it has now engrafted. It's now a member of the community. It's paid its HOA's dues and it can survive there. So yeah, we probably would have seen potentially stronger differences after a year or not if people didn't follow the diet.
It appears to me too, just based on what I read, that compliance was pretty good. And a lot of that was related to the coachability, if you will, the fact that one, they were coachable, two, that they had a dietitian calling them up 3 out of 4 weeks or thereabouts and, kind of keeping tabs and asking questions. And,
right.
So obviously that does matter. And I think, they go to the point of lifetime. Can you imagine what might ingrain as a result of that? That would be considered good, I suppose. I wanted to digress and move back a page on the study, the table 1, whereby it lists the baseline characteristics, people with Parkinson's and the control group. There, I just had a question. It was actually at the bottom of that table. It says part 1, part 2, part 3, part 4. Were those relative to weeks? Is that what those parts equal? Or what are they?
Good question. So the MDS-UPDRS, or the Movement Disorder Society Unified Parkinson's Disease Rating Scale is what that acronym means. It is a scale to measure someone's severity of Parkinson's symptoms. So if you've ever gone to a movement disorder neurologist, especially here at UF, they will do the scale with you typically every time that you come. Now, the scale actually has different parts to it. So you have part 1, part 2. These measure non-motor symptoms of Parkinson's. Okay. So it's typically a questionnaire that you might fill out. The neurologist will fill out as well. Part 3 are those classic motor symptoms. And part 4 are more dyskinesia type symptoms or motor fluctuations that someone might have. So we wanted to characterize the severity of our group's Parkinson's symptoms at baseline. So that's what those numbers mean.
There was nothing subsequent to after the study, after 5 weeks?
So we actually, what's not reported in the paper, we did actually measure.
In a summary table? Is that where that is located?
We did measure their scores after 5 weeks. However, what we realized was that, so as you all know, you will have on and off times with your medications, right? So these scores will dramatically be different whether you're on your medication or off your medication. And so within those 8 people, some people were always on their medication when we did this measurement. Some people were off when we initially took it, and then on the second time, we're on the second time and off. So that second measurement that we collected really wasn't a valid measurement.
Gotcha. Too much dynamic. Yeah,
I would have loved, but again, that's why we do these pilot studies, is so that way we can look at what should we do differently for future research and what mistakes we have made. And so that was definitely one of them. Like, okay, we didn't control for making sure people were on their medications when they took this measurement.
When I read those terms, I was thinking, regardless, because people that are medication compliant, they could still have an off period.
Exactly.
So, there's that dynamic as well, but that's kind of how I viewed it. But what you just said made greater sense with respect to the study, and why you couldn't get it what you needed when you needed it.
Yeah.
One of the things too, and I had to do, and can't remember what table that was from, but the number of bowel movements, which there was statistically non-significant, but it appeared to me as though that the number of bowel movements per week went from 6.2 to 7.5. when you even run the numbers backwards into each other like that, you're looking at 21% more bowel movements. Wouldn't that be considered significant?
Yeah. So remember, significance is really just a statistical value that determines, like, did you find this observation by chance, or is this something that we would normally potentially see? And so we likely just didn't have enough people to really
Gotcha.
say that this was a significant value. But correct, if you're an individual and you're like, wow, I could have one more bowel
every day
a week.
Instead of 6 days a week, I want 7 days a week.
That's amazing. So yeah, it's kind of like taking that's like some limitations for reporting to and collecting data that we have, as well as, that we're looking at a population as a whole. And that also means you could follow this diet and not have an increase in bowel movement.
Right. Well, I just kind of associated it with the fact of the type of foods. You're now eating more fibrous. Mm-hmm. So, I made that distinction, even though, like you said, I guess the numbers need to be bigger.
Yeah, you will see more people probably. I will say, though, it was sad I wasn't able to capture it with the stool form scale, which is that Bristol stool scale. But anecdotally, talking to people who are following this diet, it really is variable depending on the person. So, for example, I might have someone that be working with someone that has an issue with frequency. So they're only having maybe 2-3 bowel movements a week, right? And then they also have an issue with form. So when they have a bowel movement, they're rock hard, dry. It just feels like incomplete after they finished, right? So I might have some people that respond to a Mediterranean diet as it helps improve their form of the stool. So maybe they're still going 3 times a week, but now it's a lot easier to go to the bathroom because
they
now
feel evacuated. Yeah.
And then I have other people who are more frequent. I will say the more frequent are probably going to be more males.
They may not have anything to do with the BM, however, that might just be reading the newspaper.
And you bring up a good point. It could just be the individual food items in their diet that might be influencing those changes, because we know different types of fiber actually affect our bowel movements differently. So insoluble fibers, those are foods found in, think like leafy greens. So like kale, roughage, right? Like spinach, cabbage, all plant foods will have a combination of insoluble and soluble fibers, but, these types of foods will have more bran, for example, found in cereal, a lot more insoluble fiber. How that works is it irritates the lining, those cells along the GI tract, puts in more water, and it helps push stool out. So that's where you might get a better frequency, right?
Right.
And then other types of fiber, like soluble fibers, think like oats are a great example. For example, Cheerios, other types of beans, that can actually make the stool a little bit more viscous and gel-like. So that way it's a softer and easier to pass. So it probably, why people have these individual changes probably has a lot to do with the foods that they're choosing.
Right. I need to move it along because we need to wrap it up, but I got a couple more things I need to touch with you on. One of the things that I noticed was that there was a pretty substantial statistical weight loss. And I know that it made reference to that in the study because they wanted to be careful with regards to saying, hey, this is good for people with Parkinson's that experience constipation. However, it also may lead to potentially malnutrition of some sort. But when I looked at it too, I think that would almost have to go into the category of what we talked about, these microbiome that had actually increased, that looked better on the scale. Would it not be the same for weight? Who knows if they were into the Mediterranean diet now for 6 months? Yeah, the first 5 to 6 weeks, they lost 3% of their body weight, but then it just leveled off. Is that the case? Or are they going to continue to lose weight? So is there any input along those lines?
So I don't know exactly refer to the microbiome, but in terms of just like weight loss and weight changes, I think in my experience working with neurologists and in other people's experience that I've talked to, especially in neurologists themselves that I talk to, when we discuss diet and a healthy diet for Parkinson's, if you as someone went to their neurologist and say, what diet should I follow? I have constipation, I have Parkinson's, what should I do? It's, without a lot of information, it's very easy for a neurologist to say, well, a Mediterranean diet, right? It's healthy, it has fiber, it's going to help your bowel movements, right? So the purpose of pointing that out was to really inform. This is a neurology journal we had it published in, right? So neurologists will particularly read this. So we want to inform physicians, neurologists, and other healthcare practitioners that, hey, you need to think about weight before you're prescribing this to someone as well. It can't just be this catch-all diet for everyone. I'll give you an example of one lady individually in the study. She was at a lower BMI, already had difficulty gaining weight because of her Parkinson's symptoms. So a 5 to 10 pound weight loss for her is dramatic. Compared to I had another guy, more obese, enjoyed a couple beers on the weekends, right? Like classic standard American diet. A 5 to 10 pound weight loss is like him just going on a jog.
Not eating a Mounds bar at night.
Yeah. So, there's these dramatic differences and weight changes that people within the Parkinson's community can experience. And so it's really important for healthcare practitioners that are discussing a Mediterranean diet to think about someone's weight history and weight changes before just, blanket telling everyone, you got to follow this diet to be healthy, right?
Where is this study going now? Let me just ask you this. The study the paper that was driven from the study was done in 2019 in over a 5-month period of time. I think you and I didn't really even start interacting about this until 2020 at some time, right? A couple of years. And then we started to promote, hopefully try to get people more involved so you get those numbers up to the degree that you could afford to handle that, or people would be willing to travel. But so what has happened since? I mean, has to have been more people in the study since then. Now, is their data conglomerating on top, or is it becoming a separate study?
Yeah. So, since then we've started off a separate study. So not these people as well, but an entirely separate group of people. Because now we had a lot more information on what direction we should probably take a randomized control trial. So now we actually have a randomized control trial that we have concluded in June. Some of you that are listening probably have heard from Vince or myself or my undergraduate students to try to recruit for the study. But yeah, we started that study in the beginning of 2021. As I was, working on this paper already. And with that study, we implemented a control group. So they had to be willing to either follow a Mediterranean diet or not follow a Mediterranean diet.
What were the control group? Were they people with Parkinson's or not Parkinson's?
They are people with Parkinson's, and the control was the more challenging thing to distinguish.
Yeah, how do you do that? Yeah.
Right. Like, how do you control in a diet study, especially when looking at the microbiome? We know if you administer a diet, you're going to administer any form of change, you're going to see change in the microbiome. So when it comes to what's best ethically and what's best practice in research, typically when you're doing an intervention, you want to use a control of what we know that works, right? So what we know that works for constipation management is standard of care, taking laxatives, maybe increase. They could change their diet. We just were not educating them on a Mediterranean diet. So increasing their fiber. They could take laxatives if they needed to. And the control group got a handout, and we just followed them over the study intervention as well. So no
such thing as a double blind in that situation. It's impossible.
Yes. The only way it would have been possible is if we had a controlled feeding study.
Or that you threw a little something else in the diet that made it not what they thought it was, something like that,
Yeah.
I guess.
And really, as someone who works with people with Parkinson's on a day-to-day basis, I think sometimes what we miss in the research community is designing interventions that solve problems on a more realistic scale for people and their caregivers, right? Yeah. And so when designing this intervention, I wanted it to be something realistic that you could read. Maybe you don't understand everything in the article, but you could say, you know what? I can implement this in my daily life and see if it works for me, right? So that was why we didn't do a controlled feeding study. Well, number one, time, money, and resources. But if I just gave you the foods, of course, things will change, right?
Right.
Versus if my study design focuses more on, can I educate you and can you use your own food preferences and needs to create a Mediterranean dietary pattern at home and still see a beneficial effect, right? So that's the purpose of why we design it this way. Even though I would love to control for everything as a scientist, I want to make something that is realistic that you can do in your daily life.
Fantastic. Now, so that study that concluded in June, what are we to expect as a result of that? New data, new paper, what?
Hopefully, definitely new data, hopefully a new paper. Right now, we ended about a month ago. I'm working on compiling all the questionnaire data that we took from everyone and cleaning that up and using my statistical software to analyze that. I'm working on analyzing all those stool samples right now. So hopefully we'll be done with all the data analysis by the end of the year.
Well, how many people were involved in this portion of the study or this study itself?
Yeah, we had 36 people involved.
Wow. Can't wait to see some anonymity there, right? Stuff that is showing up that can be used for future studies. Mm-hmm. Do you plan on a future study again?
Yes. Well, as you know, Vince, I am a PhD student, so I'm not yet an independent researcher on my own, but that's why I'm getting the PhD and using this. And this is my project for my PhD. But the plan is to do what's called a post doctoral training here at the University of Florida, where I can continue learning new research methods in the microbiome, the gut-brain axis and inflammation. And so that way, then I can start doing some future work that maybe builds upon this.
Quite synergistic, I might add. Let me ask you this, because it was a question that had come up, is what about your diet? What do you eat?
Yeah.
What kind of diet plan do you follow?
That's why I came on. I was going to ask that too.
Oh, that's funny. I would say I follow more of, I joke like that, a flexitarian type of dietary pattern where I'm okay eating like non-animal based proteins. I enjoy animal based proteins. I really my diet really focuses more on variety versus and fruits and fiber and vegetables too. So, I mean, for breakfast, I had 2 eggs, some turkey sausage, and I had a half of a whole grain English muffin with nut butter.
Well, you and I have talked about this before, because I followed Dr. Michael Greger. I followed Ariel Balasso. These people are very much into raw foods, as a cure-all, pretty much everything. That's what they espouse. And obviously there's some attraction to what they're saying. I find compelling and curious, but because they're so far departed from the norm, like you and I are now talking about being involved with a diet that has practical applicability, you and I have also discussed the Mediterranean diet as being so flexible. It almost sounds as though you are following a Mediterranean style of diet, but because of the flexibility, you don't want to, reduce the joy of living, which eating is part of. And yet, I was going to ask you along those same lines too, with regards to your questionnaires. was there anything in there that was subjective? Like, how did you feel?
Mm-hmm.
Did you ask those questions?
Well, so I would say that gastrointestinal symptom rating scale is probably a better measure of how do you feel, because what it's asking is, did you experience any discomfort based on these gastrointestinal symptoms?
Well, and I hear what you're saying there, but on the same note, I know that when I'm having a better diet routine, because I go on and off, I fluctuate as well, but that I sleep better. So, and as a result of that, I kind of feel better in the morning. So those kinds of things are kind of subjective that I would apply to my eating better. It might not necessarily be true. It might be dynamic. Who knows?
Right. Yeah. So we didn't ask a ton of questions like that. I could say anecdotally, a lot of people felt better following the diet. Because if you're paying attention more to what you eat, you're having regular bowel movements, you're probably sleeping more, you're probably more satisfied with your day and your diet.
So it's almost a given, I would suspect. And let me finish up by saying this. One, thank you. You're beautiful. I'd love to have you come back. I would like to hear more about what takes place as a result of the most recent data that you've put together. I'd love to have you come out and visit us in person.
Yeah, of course.
You're witnessed saying yes, of course. So we'll follow that up. Yeah, no escape. But I wanted to say this document here, the Mediterranean diet, it's a 1-week meal plan, which was, I believe, used as the basis for people to follow in your study. And there is also another document which enabled people to diary day by day how compliant they were and what percentage, so that they could see how compliant they actually were with the diet. We are making this available to our group. It'll be on our website shortly, so that you can access and you can download it. I thank you for that. And again, you're beautiful. Thank you so much, Carley. Just have a great time every time we get together, and we'll be back together again.
Perfect. Sounds good.
Thanks for joining us, everybody. We'll see you next time.